So now we have covered that first step is the sampling. It means that you have to collect the sample. The second step is to cultivate them, cultivate in whatever form I have just explained. Okay. Also, there is a possibility, or it is how the marine microbiology field has evolved, right? The utilization of this imaging techniques, right? So it means whenever we think of taking any image, we think of the microscope, and the most common what we think is this. However, we know that the microscope was initially is was in this form, a single tube kind of a structure, right? And then we know that most of the marine microorganism has started after Antony van Leeuwenhoek using his lenses, right? But now we know that there are plenty of different imaging techniques are there. That includes light microscopy, having bright field, dark field, phase contrast, inflorescence, which is very much used in order to understand or enumerating the marine microbes, including the epifluorescence, confocal, and super resolution. And also at the same time, we have the electron microscopy in the form of TEM, SEM, and STM, and scanning probe microscopy, mostly AFM and STEM, right? Not going in detail about how these microscope works, but in general, few things which we need to understand is that electron microscope has enabled us to understand the ultrastructure of the cells and the viruses. In particular, when we talk about the TEM, that is transmission electron microscope, a beam of electron is used in an ultra thin section of the specimen, and that gives us the idea that what is there inside the cell. Okay, so that gives, that is being seen according to the density of the material in the cell. When we talk about the SEM, that is scanning electron microscopy, it is mostly used. That electron beam is mostly used in order to generate a 3D image, or mostly we can say the structures of the cell surfaces. So TEM is mostly used to study the inside part of the cells. SIM is mostly used in order to study the structure of the cell surfaces. Similarly, that is scanning probe microscope, one type, that is AFM, that is atomic force microscopy, where a tiny probe is held and that touches the surface of the object or any cell surface using the weak atomic repulsion forces, and this gives us idea about what is the make, or what kind of the surface for the cell, right? So all these imagining techniques have emerged in the past century, and then which has been extensively used to study the marine uh, microbes. A very recent one is a foldscope, which I again hope that many of you have seen, or may have at least uh, seen. If you don't have the option to check it, at least have seen it. In brief, it's a paper microscope, origami microscope, was developed by one Indian scientist, Manu Prakash, and his colleague in his laboratory at Stanford University, where you can see that everything is foldable, everything can be folded, and then you have pieces of lens in as an eyepiece and as an objective, and it performs exactly with the similar principle how any microscope works. But the advantage is that it comes in one particular instrument box, and then you can take it to anywhere. Okay. Those who are interested, I also have a full scope video on the YouTube channel. If you're interested to look at how it appears, how you fix it, how you see it, can go and then have a look there. Meanwhile, that imagining techniques is not only limited to what we have seen in the past, that to look the surfaces, the inside part of the cell. In the recent time, the microfluidics has also been connected with the microscopy, and there is a very famous scientist who has championed this particular field, Roman Stoker. Previously, he was in Massachusetts, MIT. and now he moved to Ivag in Switzerland. He is having a lab which I am very much fascinated. That's why I'm just suggesting that you can have a look into his profile, where he has been working extensively on the marine microbes, and then trying to look at uh, how the waves created by the organisms in order to attract the prey, the way, I mean, the way how the mortality of the microbes there in the open sea, right? So all those kind of imagine, I mean, what microscopy utilizing the fluidics, he has been using it. One of the video I would show you that will give you some idea. I hope that you can see my screen here. Now you can see that there are bacteria which has been quickly growing on the surface, right? So this is a video which is for the Vibrio, and you can see that how quickly it is spreading on the nutrient rich surface. Or once again I will just show you. So you can see my screen, and then you can see that how the microbes are growing over the nutrient rich surface, right? The another excellent example of the utilization of the microscope, and to see how the microbe works, is this one. So this is a cell, okay, I mean microbial cell, which you can see the movement or the swimming in the real time. So this is a magnetotectic cell. I will tell you that what magnetotectic is. But this magnetotectic cells, you can see that it is going upward against the gravity. Okay, so not magnetotectic, gravitectic. So it is going upward against the gravity, and this vertical swimming speed is close to 60 micrometers per second. Right, so you have seen that how this cell is going against the gravity, and all these sorts of studies has been performed in the laboratory in order to understand uh, uh, the ecology and the biology of the marine microbes. So that is what the whole point is, that we are not limited just in order to identify the species using the microbes, but we are also going further in order to understand that how the mechanisms for the ecology of these marine microbe works, right? Again, the epifluorescent microscopy, it helps us to enumerate the marine microbes, right? Because how it can help, it can help when we have a dyes which can bind to the DNA or the RNA. Now if the dyes which enters in the cell and binds to the DNA and RNA, we can look those dyes under the epifluorescence microscope, right? So basically you have a probe which hits on the target of so which hits on the target rRNA. That is, I mean, it is a specific to the species specific or group specific. And then once it is bind, you can have a look under the epifluorescence microscope to tell that whether, I mean, what is the diversity, or I mean, depending on what kind of probe have you used, you can tell the diversity of the marine microbes in particular environment. And this process also can be understood in a way that is fluorescence in situ hybridization using the probes. That is why it is called as fish. Okay. And a number of advancement have already happened in the fish. That one you can see that catalyzed reporter deposition, that is card fish. So what is happening in fish, if you have the plankton cells, so plankton will be having the chlorophylls or the other pigments that can give the autofluorescence in the fluorescent microscope. So if you are using a dye called as DAPI, okay, so or otherwise a more specialized dye like cyber green, you can separate it out that that what is the autofluorescence, if it is coming from the pigment of the planktons or if it is coming from the bacteria, right? So likewise we can utilize cyber green is exclusively utilized for the viruses, DAPI is used for the bacteria and other plankton as well. And then also there is a possibility to check that if the cells are alive or the dead in the filter. So if you have filtered, let's say 1 liter of water, and if you want to see that how many of there are the live and the dead cell, you can use CTC dyes that is specifically go and detects the active electron transport chain. So active electron transport chain will be there only in the live cells, and as an background you will have the DAPI, which will go and bind to all the DNA, right? So you will have a contrast where you can distinguish the live and the dead cell. Okay. Also, you can stain the DNA with the propenium iodide. It works as an intercalating agent, how the ethidium bromide works, right? Then, as I said, that there are advantages on the fishes like card fish, where you use the catalyzed reporter deposition, and it inhibits or it helps in removing the autofluorescence and the background noises. Another advantage in the imaging techniques, which has been utilized in the marine environment for close to 30 to 40 years, is the use of the flow cytometry. So, flow cytometry is a technique that discriminates the microbial population depending on the size, depending on the light scatter, and depending on the fluorescence of the cell. Right. It also helps you to get a single cell from the in the sample, and that single cell can be utilized for the genome, helping you to do single cell genomics for the marine microbes from the natural environment. Okay. However, you can see one result here in particular, that if you have a plankton and if you are using flow cytometer, then there is a possibility because of the autofluorescence in the pigment, I mean the pigments, autofluorescence in the planktons, whereas in eukaryote, I mean in the heterotrophic bacteria you will not have that. So you may use cyber green or you may use DAPI or any other stain, right? Not DAPI, cyber green or Toto or yo-yo, because DAPI is not a fluorescent dye. So you have to use a fluorescent dye. In case of cyber green, then what you will get, a separate location where heterotrophic bacteria will be coming. And then depending on pigment, you can see that how the result of the flow cytometry comes. You will get a separate fractions for prochlorococcus, for synechococcus, and for other eukaryotic cells. Okay. And also you add a beads, which helps as a internal standard, and that also gets separated in the flow cytometer, right? So this is how the flow cytometry works, right? So we have seen that imagining techniques, imaging techniques, this imaging techniques has evolved a lot from a single cell microscope to a full-fledged electron microscope, scanning probe microscope, and very recently this paper microscope, which is again a type of only normal light microscope. But then we have seen the utilization of UV fluorescence in the marine microorganism studies, and then utilization of flow cytometry, not only in order to enumerate the cells in the sample, but also to get the single cell, so for the single cell genomics purpose, right? Also, you would be noticing that there is a combination of the molecular methods and the imaging techniques. For example, here, what we are doing is that wherever there is a utilization or there is a application of RNA or DNA, that can be considered as a molecular approach, right? So in here we are using a probe against rRNA, right? So it's a kind of collaboration of two different techniques, that is a molecular method and the imaging techniques, and that is being very much used in the enumeration of the marine microbes.